Okay, so every cell in your body, right, and neurons and glia are remarkably interesting in terms of how they're, kind of organized. They have remarkable, kind of structural diversity associated with them. But every single component of this, remarkable cell, this is one I'm trying to build called a Purkinje cell, which is a cerebellar cell. Has about 200,000 distinct synaptic connections along its dendrites here. But every one of these dendrites is bound by a membrane. And, even within the cell, if we go to the soma here of this Purkinje cell, we have, a nucleus inside here that's, done in pipe cleaners. But there are also other, what we call little organs inside the cell, or organelles, like the mitochondria, which are really critical little oval-shaped, sausage-shaped little organelles, that actually spew out a very critical chemical called ATP. That's actually when you breathe in and you consume glucose. A lot of that glucose and oxygen end up in those little organelles, that are known as mitochondria, where cellular respiration takes place. And the sugar is, in effect burned to generate, carbon dioxide, but also this molecule called ATP, which contains within its bonds chemical energy that can be, transferred, to another form, according to the laws of thermodynamics. But every one of these little organelles, the ATP generating mitochondria, or the nucleus inside, or the cell body or soma membrane, or the actual dendrites, they all are bound by a membrane, which is, composed of, primarily these chemicals that are known as phospholipids. And they're important because the phosphate head, right, is negatively charged. It has a charge associated with it. And then there's a long sort of lipid, which is another word for kind of a fat, a tail portion of that molecule. So it's got this negatively charged phosphate head and an uncharged fatty kind of tail. And if you take a phospholipid, that sort of basic structural unit of all membranes, the mitochondrial membrane, the nuclear membrane, the dendritic membrane, the axonal membrane, the cell membrane, and you throw it into a charged environment, which is basically our, bodies, right? We have lots of water and, polar molecules. There are lots of charge, lots of ions we're going to see, like sodium, potassium, and chloride, things like that, just sort of floating about. That uncharged fatty lipid tail of the phospholipid molecule is not going to like that. Because if you, take something uncharged and fatty like oil, right, you mix it with water in your kitchen, you'll get, very distinct layers, because the charged environment doesn't care for the uncharged and vice versa. So if you take those phospholipid molecules, which have a negatively charged phosphate head and a non-charged lipid fatty tail, and you put them into a charged environment like your brain, they're going to spontaneously and rapidly form what we call a bilayer. And so that means you're going to have the charged, phosphate heads, the negatively charged heads, oriented to, the watery internal and external environment of cells. So if you have a cell membrane like this, well, the outer portion is going to be those phosphate heads, those molecules, and the inner portion is going to also be phosphate heads of those molecules, and between it will be this lipid bilayer, where the fatty tails are being isolated away from all that charged stuff on the outside of cells, what we call the extracellular side, and the inside of cells, what we call the intracellular side. So your membranes, again, of the actual neuron membrane, but also all the internal organelles, like the mitochondria that spew out the ATP, and the nucleus, which is where the DNA, the chromosomes are found, basically is bound by a phospholipid bilayer. And phospholipid bilayers are terrific. They're great as membranes because they keep certain things on one side of a membrane and other things on the other side of a membrane. And the inside of a cell is going to look very different than the outside of the cell. But we're going to see that there's going to be a lot of activity across cell membranes, like various, constituents of the environment we're going to see, including, charged substances or particles called ions, like a sodium ion, which is a positive charge, or a potassium ion, which has a positive charge, or a chloride ion, which is a negative charge, will need to be distributed across membranes, and under certain circumstances they're going to have to get across membranes, but they're charged, right? And the phospholipid bilayer has that lipid interior that's not charged and will not let stuff easily cross. The fancy word for the inability of something to cross a membrane is impermeability. That membrane is then impermeable. Phospholipid bilayer is impermeable to the passage of charged particles. So in order to get these charged particles to go back and forth, we're going to need special machines that are generated from instructions in your DNA in the nucleus, right? The expression of these genes is going to translate into what we call protein machines that will be embedded in the phospholipid bilayer. They'll actually sit there in the phospholipid bilayer with portions stuck out into the extracellular space and some portions stuck into the intracellular space. And some of these we're going to see are going to allow for the passage of charged particles back and forth. A number of different types of protein machine are particularly important for cellular and in particular neuron and glial structure and function, and these include what we call channels, which are what they sound like. It'll be a protein that spans the phospholipid bilayer, but it has a hole. This is the outside here, this is the inside of the cell. The phospholipid bilayer is in between here. This will be, there'll be a hole that will let, for example, sodium to pass through, or potassium to pass through, or chloride to pass through, or calcium to go through. Oftentimes these channels are selective. They only allow for certain kinds of charged substances to pass across the impermeable phospholipid bilayer membrane. These channels we're going to see can be non-gated, which means they're just open all the time. They have one sort of structural form, or all the structural forms they can take will leave a pore or a channel to allow for charged substances to pass. But they can also be we call it gated channels, which means that under some environmental conditions, and we're going to be talking a little bit about those soon, you can have a specific conformation where there's no pore and no hole in this protein channel, this gated channel that spans the phospholipid bilayer from the outside to the in. But then under other environmental circumstances, the structure of this particular gated channel can shift, and it can ungate and allow for the passage of, selective passage of ions, through its structural form. So there are both gated and non-gated channels. That's one sort of class of protein machine that can allow for some level of permeability across the, largely impermeable, phospholipid bilayer membrane. There are also other kinds of machines we're going to see that are really critically important that allow for the grabbing onto something from one side of a membrane and the forcing, to the other side. And usually this is forcing against some kind of natural force. And we'll talk about some of the natural forces that are involved in, shifting ions around or moving, substances from one location to another. But if you're fighting one of those natural forces, if you're actually trying to get something like a sodium ion and shove it, across a membrane to where it wouldn't normally flow through a channel. It would normally flow the other direction, but you want to force it to the other side. A special protein machine called a transporter is, basically employed. And a transporter, it implies active transport, like forcing against, some sort of existing natural force. So you need to put energy in to, basically force that, against this natural force. And so those, ATP molecules I mentioned earlier, that stored chemical energy, from the burning of sugar in the mitochondria, there's going to be a special place on that transporter where ATP can come in and a bond can be broken, and that chemical energy that was stored in that bond can be, translated or converted into kinetic movement, the shoving, of, let's say, a sodium ion, from one side of a membrane to the other, where it wouldn't normally go because of, the way things are arranged, at that time. So The other class of machine, another class of machine, protein machine that's really important that's found embedded in membranes are what we call receptor proteins. And receptor proteins are, span the membrane typically too, with the component extracellularly, lots of amino acid protein components sticking out, and then, a lot sticking in. And receptor proteins actually receive chemicals, like chemical messages. We're going to see at a synapse where one neuron, let's say the axon terminal of one neuron meets the dendritic membrane of the next. There are going to be lots of these, receptor proteins that are embedded in the phospholipid bilayers of both the dendrite and the, axon terminal. And they're going to be able to receive or listen to the chemical that's going to be released to allow for communication, from one neuron to the next at a synapse. And one of the cool things about, the interactions of chemicals, like if one, chemical, let's say a neurotransmitter that's released from this axon terminal, let's say dopamine is released, and then there's going to be, a receptor, a dopamine receptor embedded in the, the membrane of the next cell. Well, it can bind the dopamine transiently, temporarily. It'll bind it. So this is the receptor here. Here's the dopamine. They bind. And then there's going to be a shift in the structural shape, in the conformation of that, receptor protein, dopamine complex. And that structural change will lead to a functional change, in that receptor. Something will happen then. Maybe a chemical will then be released on the inside of the next cell, or, there may be a pore or channel open to allow for ions to pass. So, receptors are going to be really critically important protein machines that we're going to be talking about a lot in reference to chemical communication between neurons at those, gaps, where they meet, that are known as synapses. And finally, the last class of class of protein machine that's really important. Some of these are actually embedded in membranes, and some of these are more, autonomous or independent within the cell. Are what we call enzymes. And enzymes can either, grab onto several things and stick them together and make something bigger or, different. Basically synthesize some other chemical. These are called synthesizing enzymes. Or they can grab onto something and rip it apart. These are called degradative enzymes. So I want you to think about, again, the membranes of neurons, of glia, of all the organelles within neurons and glia, like nuclei, where the DNA is located, or mitochondria, which are burning sugar in the presence of oxygen to generate, ATP, which can run some of these protein machines. All of these structural forms are maintained to some extent by an impermeable phospholipid bilayer, made of these phospholipid chemicals that have a negatively charged phosphate head and a non-polar, non-charged fatty tail. But in order to allow for, the transit of, various substances across these, membranes, which is essential, we're going to see, for how neurons communicate, how they, send electrical messages, how they, receive or interpret chemical messages. They're going to be protein machines embedded in these, phospholipid bilayer membranes. And these include, again, channels, both gated and, non-gated, transporters, which utilize ATP, to force, things across membranes against where they'd like to go. Receptors, which are going to receive, chemical messages, change their shape, and thus functionally what they're, doing at that moment. And also enzymes that can, both synthesize and build up, substances, including neurotransmitters, we'll see, and also degrade or break down.